I want to remind you that on Tuesday we quickly covered the structure of the sarcomere. And I want to remind you what the sarcomere is actually. Sarcomere consists of two major contractile filaments. The myosin filament, which you can see right here, it's thick filament. It consists of the protein myosin, which myosin protein as the structural component, the tail, and the actin binding and ATP degrading component, the head. There are 4 heads, 2 tails. As you can guess from the functional description sorry, of the myosin head, it binds to the actin. So actin is another contractile protein. It forms thin filaments that look like a chain of beads. The globular actin forms fibrous actin. And actin molecules are bound to the proteins called tropomyosin, that's the beige line. Tropomyosin regulates the binding immediately, directly regulates the binding of actin in myosin. And troponin, that little yellow spot here, is actually, it's a protein that initiates muscle contraction. Okay, it actually starts it. It binds calcium. So if we would go back again for a second and look at the structure of the sarcomere, I'm kind of reminding you. You will see thick filaments in the center. Thick filaments are connected to each other by the M line. It consists of the protein myomycin. Thick filaments overlap with the thin filaments shown in blue here. Okay, and thin filaments are attached to the Z discs. Z discs mark the ends of the sarcomere. So sarcomere is the structure between 2 adjacent Z discs. When muscle contracts, actin filaments slide towards the center of the sarcomere, towards the M line. Does that make sense? And sarcomere shortens, and therefore muscle shortens. The A band area where myosin and actin fibers and thick and thin filaments overlap is dark. I band, the area where there is no overlap, is light. Correct? That's why skeletal and cardiac muscle both look striated. And by the way, smooth muscle is not. Speaking of which, what does it tell you about the contractile mechanisms of cardiac, skeletal, and smooth muscles? Cardiac and skeletal muscles are both striated. They both have sarcomeres, right? Smooth muscle is not striated. So what does it not have? Sarcomeres, which means they're still sliding filaments, but they are organized differently in smooth muscle. We good? So you can look at if you see those similarities in terms of the structure between 2 muscle types, that definitely says, okay, these muscle types should have, and indeed, cardiac and skeletal muscle have practically identical mechanisms of contraction, which actually will be very easy. We're not talking about cardiac muscle here, but we do talk about cardiac muscle in AP2 when we actually speak about heart. And if you know this now, if you have a pretty good understanding of this now, covering it in AP2 should be a breeze, because it all comes back. Does that make sense? Now let's move on to the organelles of the muscle cell that are necessary for the contraction. First and foremost, we need to talk about organelles that have specific names. Sarcoplasmic reticulum. Sarcoplasmic reticulum in the muscle cell is the example of smooth endoplasmic reticulum. On this picture, it is shown in blue color. What is generally the function of smooth endoplasmic reticulum in the eukaryotic cell? There are two functions. No? Okay. What's the function of rough endoplasmic reticulum? With plenty of ribosomes. If you hear ribosomes, synthesis of ribosomes. No one? No. Ribosomes. What do they make? I know it's Thursday. Come on. Friday is tomorrow. What? Ribosome. What does it make? Okay. What are three major types of molecules in the cell? Okay, four. Okay, four major types. Name four major types of biological, large, huge macromolecules. Do you read the labels on the food nutrients? Okay, lipids, number one. Number two, proteins. Number three, carbohydrates. Number four, which is not listed on the nutrient label of a product, nucleic acids, DNA and RNA. So, you have four, have 25, even if you guess, have 25% chance of guessing it right. Ribosomes, what do they make? Proteins. Good. You got it. And I know that you were not guessing. So ribosomes make proteins. Rough endoplasmic reticulum makes proteins. Now it is rough because it has ribosomes. Smooth endoplasmic reticulum, like this one, obviously doesn't have ribosomes. You got it? Since it doesn't have ribosomes, what does it not make? Proteins. So we got now 1 out of 3. Well, guess what? Nucleic acid has nothing to do with endoplasmic reticulum. So we got it out. We have fats and carbohydrates, right? Well, it turns out that in the muscle, neither. The main function of sarcoplasmic reticulum in the muscle cell is to store calcium. Generally, in other cells. Smooth endoplasmic reticulum also metabolizes carbs and fats. But here, carbs and fats are not that important. It's calcium storage. Are we clear? What is the function of sarcoplasmic reticulum? Out loud. Storage of calcium. Good, because those are very basic things, but you must remember them. Okay? You have to understand, that sarcoplasmic reticulum is entirely confined within the cell. Okay? Now, muscle cell, is surrounded by the membrane as any other eukaryotic cell, any other cell in general. The membrane of muscle cell is called sarcolemma. Does it make sense? Now, the interesting thing about sarcolemma is that it forms special structures called T-tubules. I will try to explain what they are. I may fail, but will do my best. For simplicity, imagine the stick of salami. Can you? No, seriously, imagine the stick of salami. It is covered in that little film that sometimes is really hard to chew, and I personally prefer to remove it. Do you understand what I'm talking about? Now imagine that salami is pretty soft. And you take your finger and push on that film, and you make this depression, and film goes inside of the salami. Does that make sense? If you replace stick of salami with a muscle cell, film with sarcolemma, then this depression will be T-tubal. Do you understand what I just said? Can you imagine that? That's important. So T-tubal allows membrane to reach into the cell, into the depth. You see? T-tubal on this picture is represented by this white tubule. I'm sorry. Let me cover it. So that's T-tubal. Can you see that? Go ahead.
All the way around, or is it just...
Great question. It goes around each myofibril. It's not just for the simplicity, like here on this scheme on the right. For the simplicity, it is shown like just depression. You're absolutely right. The structure is more complicated. It's branching and surrounds each myofibril. But for our purposes, what we need to understand is that T-tubule allows sarcolemma, the membrane, to reach inside of the cell, and at the same time to increase the surface area, to increase the area of contact between each fibril and membrane. Does it make sense? Okay, so far. We good? You understand? You can see on the image into the left corner, right here, that each T-tubule intimately binds with the branches of sarcoplasmic reticulum. This group of structures, T-tubule in the center, we can put it in the words like T-tubule, sarcoplasmic reticulum, sarcoplasmic reticulum. This is called terminal triad. Okay? The parts of the sarcoplasmic reticulum that interact with T-tubule are called terminal cisterns. Can you see that? That's terminal cisterns. Okay? We're good? To summarize what we just said, sarcoplasmic reticulum in the muscle cell stores calcium. Sarcolemma is the membrane that surrounds the muscle cell. Sarcolemma penetrates via T-tubules into the depth of the muscle cell and establishes contact with myofibrils. It also is an intimate contact with the terminal cisternae of sarcoplasmic reticulum. Terminal cisternae of sarcoplasmic reticulum and T-tubule form terminal triad. So far, do you follow me? Why I focus so much of your attention on the terminal triad? This is the schematic representation of the T-tubule right here, and sarcoplasmic reticulum. So you can see that's T-tubule, and that's sarcoplasmic reticulum. Look closely at this portion right here. Okay, can you see it? I highlighted in yellow. This is the part. This is the part. This one. This one. That's the part where sarcoplasmic reticulum and T-tubule are physically connected by channels. There are receptors called DHPR, dihydropyridine receptors, and RYR, ryanodine receptors. These receptors regulate the calcium flow from sarcoplasmic reticulum to the cytoplasm of muscle cell. For now, sort of take-home message is that T-tubules are connected to sarcoplasmic reticulum. We got it? Can we move on? Model of sliding filaments. That should be easy because we kind of discussed it already. So how does the muscle contract? So you have thin and thick filaments. When they slide against each other, muscle shortens. Are we clear about that? If the tension that muscle produces is more than the muscle load, it will shorten. If the tension that muscle produces is less than the muscle load, there will be no shortening. Look at me. So I hold it steady. Which muscle participates in lifting that model the most? You don't have to name, just show which part. Yeah, the flexor. Okay? Does that make sense? Does it getting shorter right now? No, it doesn't. It is contracted partially, but it stays at the same level. Does it getting shorter? Now it does. Do you understand? So if I generate force, tension of the muscle, that is more than the load, muscle will shorten. If I generate force that is less than the load, muscle won't shorten. Is that clear? You can see here that myosin heads will actually in the right picture, okay? And the myosin heads will actually bind to actin, and they will propel thin filaments towards the M line. Good? That will cause the shortening of the sarcomere. Does it make sense? Now let's take a glance what happens to different structures in the sarcomere during contraction. I'm sorry. So this is the A band. Does it change? A band. Does it change? No, it doesn't. Why? Why does it not change? The size of the A band the width of A band. What does it depend upon? Say again? Yeah, but which fibers actually determine the size of the band? Thick or thin? Thick. Do they getting shorter? No, they don't. So since thick fibers don't change the length, then A band doesn't change the length. We're clear about it? Okay? Now, what determines the size of the I band? You see that's the half of the I band here. What determines the size of the I band? The thin filaments and the distance that, actually it's the distance from the thick filament to the Z disc doubled. Does that make sense? If Z discs come closer, what happens to the I band? It gets smaller. H zone, which is the area in the center of the sarcomere, where there is no overlap. Since thin filaments come closer to the M line, H zone it disappears. Now, we will try to recount it again. A band doesn't change the size. I band becomes smaller. H zone disappears. And the Z discs, because the Z discs come closer to the center of the sarcomere. Let me tell you something. You can, of course, try to memorize this statement. But if you understand the actual mechanism of contraction and know what the A band, I band, H zone, and Z discs are, you will have no problem figuring it out. And you will have the question about the structures of the sarcomere, I promise. Clear? Awesome. Now we're going to talk about the electrical events in the muscle during the contraction. First, I want to quickly mention what the motor unit is. And we'll get back together to it. Motor unit is the part of the muscle that is innervated, that is controlled by a single somatic motor neuron. Does that make sense? By the way, speaking of which, I wanted to ask you a question. The motor neuron that controls muscle fibers immediately, is that the upper motor neuron or lower motor neuron? You can figure it out. The signal, nervous signal, that controls your skeletal muscles, where does it come from originally? Come on, it's not an exam. Say something. Where does it come from? Brain. Yeah, exactly. Brain. Which part of the brain? Huh? It's the skeletal muscle. Voluntary movements. It's a part of the cortex, right? Which cortex? Motor cortex. Good. Goes from motor cortex. Does the neuron go all the way from motor cortex to the muscles? How many neurons are in the pathway from motor cortex to the muscle? 2. Upper and lower. So which neuron directly controls skeletal muscles? The lower. Do you understand why? The upper motor neuron goes from the motor cortex to the brainstem or the spinal cord. And then from the brainstem or spinal cord, lower motor neuron takes over and transmits the signal to the skeletal muscle. Does that make sense? So these two are the lower motor neurons. Do you understand? Most motor neurons that innervate muscles are multipolar. I know it brings back really unpleasant memories about central nervous system lecture, but multipolar motor neurons, what do they have? Plenty. And what do they have? One, usually. They have plenty of dendrites, but axon is branching. Yes. So they have many dendrites, soma, and then axon that will branch. Does it make sense? In a single motor unit, you will have one individual motor neuron. You can see the purple one that I just highlighted with the arrow, okay, the purple one. And as the axon branches, its branches innervate, in this example, 2 muscle fibers. The number of innervated fibers may vary actually from like 3, 5, 7 to thousands. But the main take-home message is that it's going to be one neuron and several fibers. Does that make sense? We good? That's pretty much a definition of motor neuron. The place where the branch of the neuronal axon interacts with the muscle cell is called neuromuscular junction. This is neuromuscular junction. This is neuromuscular junction. This is neuromuscular junction. Neuromuscular junction, which is essentially a synapse. Does that make sense? This synapse signal is transmitted from neuron to the cell, skeletal muscle cell. And remember, one of the properties of the muscle is excitability. So muscle cell gets excited. Action potential is generated on the membrane and the sarcolemma. Action potential spreads across the skeletal muscle cell, causes the release of calcium from sarcoplasmic reticulum, and this initiates muscle contraction. So if we will review this in the form of a flow chart, action potential arrives at the axonal terminal right here. Action potential leads to the release of acetylcholine, exactly the same way it happened in the neurons. Acetylcholine binds to the receptors on the membrane of the skeletal muscle cell. That induces the specific changes in the ionic flow across sarcolemma, leads to the formation of the action potential on the sarcolemma, and that action potential travels across the entire membrane of the muscle cell, reaches T-tubules. That action potential in T-tubules causes the release of calcium from sarcoplasmic reticulum, and calcium initiates interaction between myosin and actin, and that starts the contraction. Okay? By now, I think you may be a little overwhelmed by all those steps. What we're going to do, we're going to talk about each step separately. So you put together a picture of the muscle contraction step by step. And first, we need to talk about the events that happen at the neuromuscular junction. The yellow structure that you can see here is what? What is this yellow structure? That's motor neuron, yes. That's the, I would call it synaptic terminal of the motor neuron, okay? Does that make sense? Good? Awesome. It contains all those standard things that we use to mitochondria, okay? Vesicles that are filled with acetylcholine, no exceptions. It's always acetylcholine, neuromuscular junction. There is no other neurotransmitter used. And voltage-gated calcium channels. When action potential arrives to the presynaptic terminal, what happens to the voltage-gated calcium channels? They open. When they open, what does calcium do? Gets into the cell. And what does calcium do to those ACH-filled vesicles? Okay, influx of calcium into the presynaptic terminal. What happens to ACH? Calcium influx leads to? Releases, yeah, ACH is released. Does that make sense? ACH is released. Does it make sense? Where does it? It is released into the structure that is called synaptic cleft. Okay? So there's this, it's a space between the terminal of motor neuron and the sarcolemma of the muscle cell. You can see that sarcolemma of the muscle cell in the synaptic cleft forms folds, so-called junctional folds. What's the function of those folds? Increase surface area. Now those little tiny molecules here, what are they? The tiny molecules, blue molecules on the sarcolemma that bind acetylcholine. That's acetylcholine molecules. Receptors. Awesome. Actually, ligand-gated cation channels. Okay, so they open and they bind acetylcholine. Now let's review steps again. So you can see motor neuron. That's the individual neuromuscular junction. You can see synaptic. So the action potential arrives to the presynaptic terminal, depolarizes the membrane. That opens calcium channels. Do you follow me? Okay. Calcium enters the cell. Calcium entry causes the release of acetylcholine into the synaptic cleft. Acetylcholine binds to the sodium-potassium channels on the sarcolemma. Channels get open. When sodium-potassium channels are getting open, where does sodium go? In what? In which cell? Huh? In the muscle. Where does potassium go? Out of the muscle. But the sodium influx is more than potassium outflux. So more sodium gets into the cell, then potassium gets out. Okay? What happens to the membrane potential of the skeletal muscle? How does it change? It becomes more yeah, it becomes more positive. In terms of depolarization, repolarization, what it is? Depolarization. So, muscle cell locally depolarizes. Do you understand? Think about this. This is still local depolarization. Okay? Does that make sense? Now, remember what we need to do with the neurotransmitter after it exerted all those actions. We need to degrade it. Yeah. Acetylcholinesterase breaks it down, because what will happen if you hypothetically inhibit acetylcholinesterase at the neuromuscular junction? Huh? In terms of like macro effects, what's going to happen to the muscle? Yeah, spasms, contractile spasm, yes. Does that make sense? It's going to be contracting all the time. Got it? So far, we're good? Don't even start. No. Cramps, the nature of cramps is very complex, and currently it is thought that it's both sodium, potassium, and calcium imbalances taken together. It is rarely just one ion. That is why, people say just eat salt during the run. Nope. Bananas potassium? Nope. Some suggest pickle juice, which contains sodium and potassium. Generally, it is believed that cramps, muscle cramps, arise from all three ions being in imbalance: sodium, calcium, and potassium. Bless you. That makes sense? So now I want to remind you that local depolarization is, it's not the entire muscle, it's not the entire, it's just local. So the local depolarization is called end plate potential. Okay? Are you with me? Right now, very important thing. In the rest of the sarcolemma, right here, there are sodium and potassium channels. And these guys are not ligand, but they are voltage gated. So what opens and closes them? Can you tell me, please? They voltage gated. They open and close in response to changing membrane potential, right? And look, membrane potential just changed right here at the neuromuscular junction. If the change is sufficient to open those sodium channels first, here, okay, sodium channels open first. Sodium channels open, what happens to the sodium? It goes into the muscle cell, causing further depolarization. Does that make sense? When membrane potential of the muscle cell becomes high enough, sodium channels close and potassium channels open. Now potassium will leave the cell, causing repolarization. Does it make sense? Okay? Now, think about this for 1 second. You start with the local depolarization at the end plate, at the neuromuscular junction. As adjacent sodium channels open, the depolarization wave spreads away from neuromuscular junction. Does it make sense? Like dominoes start to fall. Eventually that wave of depolarization reaches T-tubule, releases calcium, leads to contraction. Okay? Do you understand that end plate potential leads to development of depolarization wave across the sarcolemma? Is that clear? I'm going to do a little bit like executive summary after that slide. Now let's look at the changes in ion movements across the sarcolemma. So during depolarization of the muscle cell, sodium channels, voltage-gated sodium channels, open. Does that make sense? So they enter the cell via facilitated diffusion. Got it? At the same time, voltage-gated potassium channels are closed. When cell is completely depolarized, here at the peak, okay, voltage-gated channels are inactivated. They're not just closed, they're inactivated. It's a short refractory period. Why refractory period is so important? Imagine for a second that sodium channels would be able to stay open like forever. What would it cause on the macroscopic level? Depolarization causes contraction. If sodium channels constantly stay open, muscle cells will stay constantly depolarized and contracted. Spasm, tetanus. Does that make sense? So that refractory period prevents tetanic contractions. Are we clear? Now during repolarization, potassium flows out because voltage-gated potassium channels are open. And then, at some point, sodium-potassium pump restores the concentrations of ions across the sarcolemma. Does that make sense? Action potential arrives at the presynaptic terminal, opens calcium channels on the neuronal presynaptic terminal. Calcium flows into the neuron, leads to the acetylcholine release. Acetylcholine diffuses across the synaptic cleft, binds to the ligand-gated sodium-potassium channels, which open, letting sodium in and potassium out. Sodium gets more sodium gets in than potassium gets out. It leads to the local depolarization. It is called end plate potential. Local depolarization causes opening of voltage-gated sodium channels that are close to the neuromuscular junction. Opening of those voltage-gated channels leads to the wave of depolarization across sarcolemma, action potential essentially. Sodium influx in the action potential is responsible for depolarization. Potassium efflux is responsible for repolarization. And sodium-potassium pump is responsible for restoring the concentrations of ions across the membrane. Good? I will ask you, there will be several questions on the exam. regarding the ionic flow in the muscle. So this process, sorry, this is called excitation. Okay? You understand that? That's excitation of the muscle. Right? Now we need to talk how excitation is coupled to a contraction. Do you understand the term coupling? Physiologically, chemically, what's coupling? Two processes are intimately connected. Okay? Contraction doesn't occur without excitation. And excitation will probably lead to contraction if everything works fine. Okay? So that's coupling. So eventually action potential, which is shown on this left bottom picture in the purple line, reaches the T-tubule. Okay? Now you have to appreciate that there is time from the moment nervous signal reaches the muscle to the time contraction occurs. The reason for that, action potential has to actually go across the sarcolemma. It takes time for action potential to spread. This is called latent period. Does that make sense? So when action potential reaches T-tubules, it activates the receptor DHPR, which then stimulate RYR, and calcium is released into the cytoplasm. The depth I want you to know this interaction is, I don't want you to memorize the names of those receptors, DHPRs and RYRs. Okay? What I want you to understand is that T-tubules and terminal cisternae, so T-tubule and the terminal cisternae of sarcoplasmic reticulum are connected. And when action potential reaches that terminal triad, by the system of receptors, it causes the release of calcium from the sarcoplasmic reticulum into the cytoplasm. Are we clear? Is that understood? Now first, so here's what we're going to do with the mechanism of contraction here. First I'm going to explain how it works, sort of, step by step using pictures. And then we're going to do a little play, if you don't mind. I will need volunteers. It's kind of fun. It helps you to memorize, I hope, the interaction, okay? So this is the muscle that is not contracted. Calcium concentration is low. Myosin fibers, myosin molecules are not bound to actin because those binding sites are blocked. Those little holes represent binding sites. Okay? These binding sites are blocked by the molecule called tropomyosin. Okay? When calcium concentrations increase, calcium binds to the molecule of troponin, and binding of calcium to troponin shifts tropomyosin, as beige lines, away from binding sites. Are we clear? Binding sites become available for the myosin head to bind. Myosin head binds, forming cross bridge, and that cross bridge propels the actin, the thin filament, towards the M line. Is that clear? You good? Now let me pause. Come on. Moving on. So now, how contraction is a mechanical, right? It's a movement. Does movement require energy? Yes, it does. What is the ultimate source of energy in the cell? Brilliant. ATP. So somehow, muscle has to break down ATP. When we were talking about the structure of the myosin, we mentioned that the myosin head can break down ATP. Does that make sense? So, I don't like the sequence that they have here, so I'm going to start with a step number 3. Okay? Myosin head binds ATP. Simple as that. It breaks it down into the ADP and inorganic phosphate. That make sense? When you break down ATP, this breakdown releases energy. Everybody agrees? Head, this energy kind of coax the myosin head back. Like, you know, like you... Not really good with weapons either, but when you have like single action pistol and you cock back the trigger, is it called trigger? I think it is called trigger, I'm not sure, but you cock it back, okay? So this is what happens to that myosin head, right? Myosin head then binds to the actin and releases ADP and phosphate. That release is sort of a signal for myosin head. And remember, it is charged. It is coaxed back. It stores that energy. So when ADP and phosphate are released, myosin moves forward. Does that make sense? Do you understand what I'm saying? Okay? You can think about how do you call this children's weapon when there's like fork like this and there's a rubber band and you pull it back. Huh? Slingshot, yeah. So you pull slingshot back. Until you hold it, it's not going to fire, right? So that charged, this is the charged slingshot. When ADP and phosphate are released, slingshot fires. And the actin filament, the thin filament, is propelled towards the M line. Does that make sense? Okay? Now, even after that propeller, even after that, myosin head is still bound to the actin. What it needs to bind to get released? ATP. It binds ATP, gets released again, and is ready to charge. Does that make sense? I will try to show it to you. This ruler is the actin molecule. My hand is the myosin head. Does that make sense? Myosin head binds ATP, breaks it down to the inorganic phosphate and ADP, and that breakdown charges it. Okay? Myosin head, charged, binds to the actin, releases ADP and inorganic phosphate and propels actin forward. In order to be released, it binds another molecule of ATP and releases. Breaks down ATP to ADP and phosphate, charges, binds actin, ADP and phosphate are released, actin molecule is propelled further. Does it make sense? So this is called a cross-bridge cycle. Cycle because it is obviously a cycle. Cross-bridge because the structure formed by the head of the myosin and the actin, this is called a cross-bridge. We're good. Now, when does the cycle stop? When there is no more ATP. I'm going to tell you a secret. It's almost impossible to run out of ATP in the cell because it is produced. It's practically like, it's really hard. What else? Cross-bridge, is formed by which two molecules? Cross-bridge. This is formed by what? Cross-bridge is the interaction between actin and myosin. When can actin interact with myosin? In the relaxed muscle, actin doesn't interact with myosin. Why? Tropomyosin blocks it. So actin interacts with myosin when tropomyosin is shifted away, right? What makes tropomyosin being shifted away? Huh? What makes tropomyosin? Calcium that binds to troponin. Now, so if ATP runs out or if calcium runs out. Does that make sense? So this is how we stop the contraction. Does that make sense? If we get all the calcium from the sarcoplasm, and I want to remind you, this whole thing happens in the sarcoplasm, in a cytoplasm of muscle cell, in the sarcoplasm. As soon as we pump calcium from the sarcoplasm back into the sarcoplasmic reticulum, contraction stops. Does that make sense? Do you understand? Because if we pump calcium back, this whole sequence will run backwards. No calcium, okay? Troponin cannot shift tropomyosin away. Tropomyosin blocks interaction between calcium and myosin. Boom. No contraction anymore. Does that make sense? Now, interesting condition called rigor mortis. Detachment of cross-bridge here, step 3, sorry. This step right here. Detachment of cross-bridge requires ATP. When a person dies, ATP is not produced anymore. However, post-mortem, cells start to take calcium in, including muscle cells. So calcium flows into the cells. Elevated calcium levels cause binding of myosin heads to actin. And myosin heads, they break down the remaining ATP causing contraction. But the problem is, after the contraction, they can't detach. Because only after detachment, muscle can relax. They stay attached to the actin. Muscle stays contracted. And this is called rigor mortem. Okay? As far as I remember, it starts about 5 hours, okay, after post-mortem. And this is, I mean, it is weak, but if it's a weak contraction of all muscles in the body becomes really rigor, okay? Does it make sense? It's really hard to bend. Eventually, you know, if you leave the corpse for extended period of time, then cross bridges that are essentially proteins will be destroyed by proteases inside of the cell, the enzymes. They'll just disassemble, and if cross bridge destroyed, then muscles become again completely relaxed. Does that make sense? Now, muscle contraction is severely affected by the calcium levels. Hypocalcemia leads to the hyperexcitability of muscles. Okay. Hypercalcemia leads to the muscle relaxation, which looks a little bit counterintuitive, but turns out the mechanism is not linked to the intracellular calcium, because the main source of calcium for muscle cell is sarcoplasmic reticulum. Does it make sense? Main source of calcium for skeletal muscle is inside of the cell. But calcium regulates sodium transport across the membrane. And it turns out that the lower extracellular calcium leads to the increased sodium flow. The more sodium flows into the cell, the more frequent action potential is going to be. Does it make sense? Now we're going to talk a little bit about, let me see. You know what? We're going to take a break, and we'll talk about muscle mechanics when we come back from the break. All right?